from online participant, can I hear and see my screen?
Yes, it's working. Thanks, Laura.
Okay. So, hello. First of all, very big thanks to the organizers, to Ivana, to Milos, to all the team for having me here. This is really a wonderful opportunity. I'm learning so much, so thank you. And for the online participants, today it's snowing and the landscape is crazy and amazing. And I'm Laura Gazzoli, and today I want to present to you about my experience, limited, about culture collections, from how to build them and how to manage them. And something about me, I did my PhD in marine mycology. Then I moved to the microtech of the University of Turin for my postdoc. And this is a fungal collection that hosts more than 7,000 fungal species, isolated from all the different substrates from all over the world. And there I worked as a taxonomist of marine ascomycetes, and I was the curator of the marine fungal collection there that currently hosts 2,500 species strains. And the collection is affiliated to the European Cultural Collection Organization, the World Federation of Cultural Collection, the Microbial Research Infrastructure, and also have an internal quality system certified for internal and external procedures by ISO 9001. Then I got a position as a researcher at the National Research Council of Italy. There I joined the microbial ecology group. They have a very high skills in microbial ecology and specialization in bacteria. And I'm trying to apply there my competencies on fungi. So that's why ParAqua is so pivotal to me because I'm shifting to marine fungi to freshwater fungi. And this is a very nice place to be. It's on the lakeshore and subalpine lake. We have all the competencies on freshwater and environments. We have rotifer expert, bacteria expert, and we also have a guest house for free for international researchers and collaborators. And we are very happy to have all people from all over the world to come and visit us. And there, together with my colleague Esther Eckert, we won a project with other culture collection, Italian culture collection, that is the implementation of the Italian node of the microbial research infrastructure. And we gained funds to build our own culture collection of bacteria and fungi. So I'm very well aware of the difficulties to start from the scratch to have your own culture collection. So basically what I will bring here is my experience on fungi, but these rules that I will show you today apply to all kinds of collection, from microbial to microalgal collections. Some definitions: what are culture collection? They are also sometimes called biorepositories or biological research centers. And they are facilities that have been, that collect, catalogs, and store parts of organisms or living organisms. We have collection for human tissues, for viruses, and generally they have been established for a purpose. For example, food and airborne fungi or human samples. So they have a purpose. And in recent year, there is a general discussion on how to define biobanks that are different from culture collection because they are legal entities or part of legal entities, and they have some legal framework, and they are built to acquire and store and transfer the microorganisms. And this concept has been recently formalized by the ISO 20387 quality biobanking framework in the international standardization organizations. but there is still some discussion going on about the terms. For example, should a biobank should be big or small? What are the purposes? And so these definitions are still in progress. So I will refer to culture collection for what I'm building, because achieving the accreditation for biobanks is very tricky. What are the pros of having a culture collection in place? You can have a treasure of opportunities. You can use your strength to bioprospecting, to make experiments, to have experimental trials. Also, if you have a very well-established collection, this can be a priority when you are applying for projects, for example, because you have a very organized structure there. And usually SMEs and industries are more likely to work with this kind of institution because they have formal agreement, for example, that they can read and see, and this agreement can be formalized and signed and they can refer to. So they are more happy to work with this kind of institutions. And if you have a very well-managed culture collection, you can interact and gain visibility by connecting with these global and European initiatives that we will see later. And also the culture collection gives you the possibility, if you have a third-party list of services, to self-sustain your collection and your research. On the contrary, culture collections are super expensive. They are very time-consuming. They need a lot of space for the things to be, for the organisms to be stored, and facilities, and dedicated personnel. And it is very difficult to comply with the new regulations, old and new, let's say. So what are the quality criteria? Internal, you have to define roles and procedures. You have to define training for the personnel. Waste is, for example, the waste treatment. You have to understand how to organize, to build the knowledge in there and to exchange ideas. And if you want to comply with the ISO 9001, it is even more difficult. This is my expression when I think about it. And for external quality, you have legal documentation and compliance. You need to assess traceability, credibility of your collection, accountability. So this is really a lot. I know that, but we are here today and we will try to discuss about this. So what I want to do now is to go through the processes when you want to acquire a microorganism and store and manage the collection and transfer the strains and valorize your collection. And of course, I can bring my expertise on fungi, but I'm here to learn from you. So feel free to interrupt me and ask me questions or asking me your opinion about the things that I'm saying, that perhaps I'm saying stupid things and you know more. I know that you know more on some of these things. So for acquisition, one thing that we should keep in mind is the definition of biological resources that has been provided by the Convention of Biological Diversity. So what are biological resources? Are genetic resources, organisms, part of them, that are of potential use for humanity. And what are genetic resources? They are any plant, animal, or microbial materials that contain functional units of heredity. And this is very good to keep in mind, that we will see later how to comply with regulations. Because in 2010, it comes in action the Nagoya Protocol. I know many of you perhaps deals with it, and you know how difficult it is to deal with it, because I know it is a lot to do to comply with this, but the Nagoya Protocol come into action to prevent biopiracy. There was companies that stole the intellectual property, and they made millions of money, for example, using traditional plants for medicines, for drugs, without corresponding any money to the countries in which, like, this medical plant, for example, was collected and harvested. So the Nagoya Protocol is on the access and benefit sharing and the fair and equitable share of the biological resources. So if you want to acquire or even isolate a microorganism in another country, if the country signs the Nagoya Protocol, you need to have some documentation even to just isolate this organism, if you also plan to make some DNA extraction, for example. And what you should have? You should have a mutually agreement terms documents that state clearly what are the benefits for the country in which you are isolating the microorganism. And when I say benefits, those are not just monetary benefits. They are, for example, transfer of knowledge, they are publications, they are training, but this should be clearly stated in this document. Then if this organism that you are isolating will enter in your collection, you also have to have a prior informed consent contents document called PIC, I don't know how it's pronounced in English, and a material acquisition agreement in which you have to state where you isolate and when, because the Nagoya Protocol came in place in 2014. So all the materials that you have stored in your collection before 2014 does not require this kind of documents, but if you have something stored after 2014, this might be needed. And if you're not sampling, if you're sampling in a country that is not signatory of the Nagoya Protocol, you need to have to contact local authorities to find, to be sure that you are complying with the law. And so once you have all these documents in place, you can start isolating your microorganism like we did today. So with serial dilution, direct plating. I was working mainly on microalgae and no, I'm with algae and macrophytes, so I can bring some experience on that, but of course there are several other methods. Serial dilution that we have seen yesterday, as Silke said also yesterday, is very convenient because you can put into your collection strains and assure of monoclonality of the strain. That is very important. And also because some, for example, marine fungi are sensible to antibiotics. I know it's strange, but it is difficult. So for some groups, if you put antibiotics in the medium, they are difficulties to grow. So these methods that we have seen yesterday, it is very cool. No? You can try also different media, mimic one, rich one, poor one, and different temperatures. For marine fungi, for example, they grow better at 3.4% of salinity. And you should record all this kind of information also on culture media and temperatures. Because you have to be sure to have all the possible information, so data and metadata associated to your strain if you want to put it into your collection. Also, you should record information on how you identify the strains, because methods can change in time. If you will have your collection that will last forever, like in the future, and the taxonomy methods that we use today are not suitable for 20 years in the future. And for example, for fungi, ITS is not always the best marker to have an identification at species level. So you should mark down what is the marker that you use for the identification, and also blast results, on which blast adjacent number you base your identification. You should store also ABI files, because I think that probably in the future GenBank will ask for them to check the reliability of your sequences, and pictures, of course. And how you organize this information. Usually you have all your experiments and you mark down the things that you've done, your strains. But if you want to organize your culture collection, you need to pass from this kind of story information to these massive data sets about your strain. And it is an enormous table. And why so enormous? Because if you want to valorize your culture collection, you need to have what is called a minimum data set. A minimum data set is like an Excel file or MySQL file should contain all these fields. For example, the geographic origin, the pathogenicity, and all this kind of information that certify that your strain has very good associated data. And you can have it on an Excel file, or it is better for users if you have multiple teams working on your collection to have an access file or a website in which the operator can put all these informations on acquisitions. What are these data? Taxonomy and isolation are what we said. Non-growth media, for example. Some strains cannot grow on very generic media. They require particular growth media to survive. And what are the sporulation conditions, for example? In which media you can see the anamorph or the teleomorph, and what temperatures it can survive. And if you want to organize your collection to work with SMEs and companies and to have your strains accessible, you should note down if this strain is free for unrestricted use or if it's patented. Because if it's patented, you have an agreement with the industries and you cannot distribute this strain, and all the operators should know about this kind of information. And also if the species, if the strain is a type species, so it has a particular value, for example, for nomenclature. You should note down the biosecurity level associated with this strain that we will see in a moment, and if the strain can be used for dual use. The dual use means that these strains have the potential to be used for military purposes, for example, anthrax. And if there are culture collections that store this kind of microorganisms, this should be stated, because if you want to transfer this strain, you need to have specific approval from for the military use, to distribute a potential threat. And what are the preservation methods that we will see, and all the legal documents associated. And for these fields, I should have like the name of the columns should be according to common vocabularies that have been decided among the scientific communities. For example, the Darwin core thesaurus is a vocabulary, control vocabulary. And why you should have this, like the name of your column so standardized? Because if you put your catalog online, data repositories can access your catalog and show the information about your strains in their repositories. So they need to know where, like for example, the substrate of origin is substrate and the substrate origin. And so the machine can know that in that column you have all the information about the substrates of origin. And it can show, like for example, on the World Register of Culture Collection, on GBIF and so on. Biosecurity level. In 2000, a regulation came into place that defined what are the biological agents. That are all those microorganisms or part of them, or cultures or endoparasites or viruses, for example, that might harm humans. And in this directive, they are divided into groups, from group 1, that is not likely to have any impact on human health, to group 4, that are biological agents that cause severe disease, that cause a serious hazard to worker, that present high risk to spread to the community, and for which there is no effective prophylaxis. For example, COVID-19 is a group 4 bioagents. But if you're using not like fungi, bacteria, there is a list in which you can refer to understand if your strain is among this group. And please note that if your strain is, for example, multi-drug resistance, it might change group. And for ParAqua purposes, the directive states that animal and plant pathogens that do not affect man are excluded. but notes that even plant pathogens, for example, or animal pathogens, should comply with other regulations, for example, on pest control or on agriculture. I'm thinking about Xylella fastidiosa. I'm thinking about some animal disease that if you want to transfer to another country, you should comply with those regulations. So this information should be noted if there is known pathogenicity. Also because if you will deal with this kind of biosecurity level, see the WHO, the World Health Organization, have listed some fungi, for example, in the list of priority pathogens, among which there are some fungi that are commonly retrieved as airborne, like Aspergillus fumigatus, like Fusarium, like Mucorales, Saccharomyces. So if you're dealing with if your operator is dealing with this kind of microorganism, there should be biosecurity levels in your laboratory. So equipments that are made for protect the worker and to prevent the spread of the microorganism. This is a biosecurity through laminar fluid hose, and then for biosecurity for group 4 agents, you have, you know, that fluid hose in a specific room with no access. You have to have the contamination when you enter and go out. And so, of course, you have to train personnel not to do like these two weirdos that are playing with the cultures collection and without knowing about the risk associated to microorganisms. So you need to train your personnel or prevent them to do these stupid things or to access to part of your laboratories that are not accessible if you're not trained. And of course, warning signs. Any questions, Antonio? So how do you store your strains in culture collection? It is advisable to have different methods to preserve your strains. At least 2 methods. I'm talking about bacteria and fungi. I don't know much about algae, but I think they can be cryopreserved too. I don't know. Probably. So about active growth, of course, each of your strains should have a code that's linked to the catalog and the associated data. For active growth, these are part of the collection into a refrigerator at 4 degrees. And the collection ideally should be in in a room with limited access. For example, here we had a code to access this room, because like students or technicians that do not deal usually with this kind of strain can like take the tube and make copies and forget it somewhere, and then the strain dies. So like in Turin, we had a form in which the students or postdocs wrote down what was the strains that they wanted to access, and the responsible of the collection make the copy and give the copy to the personnel. And so just the responsible of the collection have access to the active growth collection. And of course, you should have different, like refrigerators should be different and incubator should be different. So to prevent some unintentional break or lose on your collection. And active growth is good for, it is nice for fungi. For bacteria, you might have problems with active growth because of recombinations. So you can lose with active growth the genetic peculiarity that were associated to the strains because of recombinations. Lyophilization for fungi can work good for sporulating fungi. You should have equipments for freeze drying your strains that maintain the sterility of your culture. And it is better to have the lyophilized and the active growth in different rooms or even in different buildings. Because if something happens, like a flood or a fire, you have a backup plan. And this is true also for cryopreserved. That works good for bacteria, for fungi, I know that some cyanobacteria experts are using cryopreservation. And very briefly, what you do is to grow your fungus on a media that is rich in glycerol. So it accumulates glycerol in the cell. Then you cover with the glycerol and how you say? You have this small straw and you put plaques of mycelius in there and you seal them. And then you have to lower the temperature, ideally until minus 152. But of course, at Microtech at CNR, I just have a minus 80. And some use you should very fast lower the temperature to prevent the formation of big crystals. So it is, if you have liquid nitrogen, you can use liquid nitrogen to like deep freeze very fast your strain. But the ideal procedure is to lower very slowly the temperature until minus 30, 40. There are some equipments that can do that, and then deep freeze as minus 152 to keep the cell integrity. And like here you can see two, like we in the Turin we have two minus 152 that stores two copies, one in one refrigerator and one in another refrigerator. The refrigerators at minus 152 were on a separate electricity line. This electricity line has an emergency generator, and this have also an internal battery that if all the electricity go down, they activate. And we had an internal system. There are data loggers that can resist to this temperature with a SIM card that can call you and say, oh, something is going wrong with your collection. But of course, this is super costly. Mm-hmm. So what I do is I freeze at minus 80. And also for fungi, one very old way, but still effective, is to use paraffin on your tubes, sterile paraffin. This is a strain from my PhD thesis. It was in 2012, and I reactivated it, and it still worked. Not for all the fungi, it's very cheap. And it's not the best, but if you don't have refrigerators, this can be an alternative. And about management, of course, you need to define the roles, so responsible of each room, for example, and cleaning a calendar, because sometimes your collection, like your laboratories, become a mess. If you ideally in your database, you should write down when you transplant the strain, or when did you perform cryo or live preservation, and periodically you should check for cell viability. So if your strain is still viable, and you need to be sure that the technician, for example, that is responsible for transplanting is doing it periodically, so you don't lose the active growing strain. And of course, we have seen it is better to have a backup of the database on the website, but even on a hard disk that you bring home when you go home, because sometimes, like, we had burglars that stole the server. And so fortunately, we have this backup plan at home of an hard disk. And you should have also a disaster plan. What is a disaster plan? If a fire or a flood appear, you can have all the generators and data loggers and warning systems. But if a fire happens, you should have like a backup of your culture somewhere else, for example, for cryopreservation. But ideally, to have multiple copies in different collections. And I know this is not something easy to do, because you should really trust the owner of the collection that will advise you whenever he wants to do something with your strain. And for type strains, for example, for bacterial type strains, when you deposit a new name, they ask you to have your strain deposit into collection for bacteria. And now they will ask you to do this for fungi too. So about transfer, if a third party, for example, an SMEs or a company is asking you a strain, you should have an agreement on that. That are called MTA, Material Transfer Agreements, that are in respect to the Nagoya Protocol, so the access and benefit sharing. And in this document, you should state very clearly what this company or third party will do according to the due diligence principle of the preservation of biodiversity. And you should state if the strain is patent, all the terms for use, and all this kind of information. About valorization, you have done all this work and now you can have fun with all the strains that you have. You can do whatever your imagination can imagine. You can, for example, see if your strains is a marine strains that, no, not a marine strains, but if you grow the same fungus on different media, it activates different metabolic pathways. So you can have different metabolic profiles. For example, you can do mesocosm, microcosm. I'm thinking about algal experiments. You can also test for bioprospecting for enzymes. But you should record all this kind of information because, for example, a company arrives and asks you for lac cases that is able to work at high salinity. You should fastly know what strain you should look for. So this strain can grow at this salinity at this temperature and can produce this enzyme. So in your data sets, you should record all those, also this kind of information. And to valorize your collection, we have seen that you can have a list of services. Like, for example, you can provide identification services. We were working for a very famous company that was trying to remove alcohol from sweets to access like the other markets. And we provide these identification services with an agreement, and this gain us a lot of money. And this is because we have seen it's so costly to have a culture collection, so you need to have some resources dedicated to the collection. And this is a good way. You should have a website or at least an online catalog that needs to be interoperable with the other how to say, initiatives. And briefly, what are World Federation of Culture Collection is, let's say, an agreement. Federation of Culture Collection currently hosts more than 700 culture collections. And the aim was to have, to collect data about the strains that are collected worldwide and make catalogs of the strains, of the type strains, of the services, of the data, of genomic data. So they are not very modern website, I have to say, very difficult to navigate, but very useful. And when I started to think about my collection at CNR, I brought to the World Federation of Culture Collection what I have to do to be interoperable with you. And they answered me that affiliates should be qualified to certain standards. And they give me a PDF document that I have to fill, in which I should say if the collection is independent from my expertise. So if something happened to me, I will stay in the grade and will never be back because it's so beautiful. Someone else can take up my work and continue the collection to avoid the risk of orphan collection. If I am willing to undertake the work to have my catalog interoperable with the World Directory of Culture Collection. And this is true also for the parallel organization of Europe, European Culture Collection Organizations, that has a catalog of strains that has these minimum data sets that can be visualized of over 305,000 strains, and not just fungi or yeast, but phages, plasmids, and also I think plant cell and algae and protozoans are accessible to the ECO catalog. And even in this case, your collection should provide public service on demand and without restriction, accept cultures for deposit and provide catalogs. And so a lot of work to be done to interact with them. And brief discuss about MIRI. MIRI is an ERIC infrastructure. What are ERIC infrastructure are research infrastructure that the European Union plan to capitalize on. So they are investing a lot of money in the maintenance of this research infrastructure. And when the European Union make some calls for projects, they are asking to us to interoperate and interface with these ERIC platforms. And in the vision of fairness, they want projects and the results of projects on biological resources to be accessible through this research infrastructure. And through MIRI portal, you will see there are some nice tools. It's just not microbials for researchers. There are tools for industries, for data research centers. There is a list of services, a list of consultants for the different strategic areas. For example, I've browsed for algae, and they provide services and collection, for example, for microalgae, for bioactive compounds, foods and feed and biosec. So you can have fun there. You also have the pairwise ID possibilities, and you can browse the catalog of collection associated to MIRI. And this is why it is very important to have these minimum data sets, because your strain can be visible in this catalog if you have these minimum data sets. If you are super interested in biobanking, there is a very nice and beautiful course by the Institute Pasteur on biobanking, not just on fungi, but on biobanks of bacteria and viruses and human tissues and a lot of things. It is freely accessible through the website. But if you pay 150 euro, you can also have a certificate of attendance, and it's closing on May 12th. So if you are interested on the topic, you have all the videos and materials. It is very fun. And that's it for now. It takes a lot. This is my email. And I'm really looking forward to interact with you in the roundtable if someone will still be here, because it's 5 in the afternoon. I know many of you will. So in case, this is my email. And thank you for your attention.
Just a curiosity, how was in the pandemic situation, the activity in the
you could have special permissions, like the one that you have for work, to go and manage the collection. Like if you were an independent worker, you can sign off yourself that you can go there. And our director was signing this special permission to go there and access the culture collection. Of course, I'm expecting that after COVID, things might change on culture collections, especially on biosecurity, because there will be some big changes in the biosecurity level requirements for organizations that are doing biobanking. Of course, because, like, for example, animal pathogens that do not affect man are not listed now, but for the transmission of pathogens, something might change after COVID for a culture collection. For the accessibility, we do not have much problem.
Told us that she came back from the US with some tube of algae.
No, she never told that. Do you have to?
No, well.
She never did.
Anyhow, what is the procedure in order to ship some biological material?
No, for the shipment, you should have the clearance for content and the material accession agreement. For have this, like this strain store in your country. If you want to access some materials there, of course, you can state that you will not extract DNA and you will not use this genetic resource, because the Nagoya Protocol is on genetic resources. But of course, you should have legal permission also for quarantine purposes, because they will ask you if the strain might be harmful for animals and plants. And for example, in Italy, there is some projects going on with the police of the borders, especially on the airplane, to prevent the spread of invasive species, for example. Because microalgae, I'm imagining there are some species that might be invasive in the new environment. So to prevent the spread of invasive species, there are some border stop on some species.
Maybe I can add something unique because of international trade, and you need permission from ministry for some strains, for some not actually, but you have to ask the Ministry of Agriculture if you need permission for that, and it depends on country for which you need. So we have permission. Phytosanitary certificate, and this law.
So it's complicated.
It is, because when you come at the border, when you ask another country a certificate for algae, this is a bit strange, but it's normal for all plants. And then they can keep you in quarantine, and this is what I was worried for actually, because what they will keep it 5 days and think I will get permission at the end, but what will happen? Will open my algae to get some CO2 or something. So what will be at the end?
Thank you.
Yeah.
What is that you need to keep this collection, the collection of this size?
The one in Turin, they have now they have 3 technicians. One is dedicated to the active flow and management, ISO management, because they will also have this internal procedure, and you have to are responsible for internal procedure. And one for lyophilization, one for cryopreservation, and postdocs. We have 2 professors that are now managing the collection, and for all the external research, they now have 7 postdocs. But let's say at least, like with this project that have been found, I will have 1 technician for the collection and for the management. And ideally, you should have another technician for the catalog that has also training in taxonomy, because ideally your catalog should be one accession form for students, for example, or industry, that with basic data. And then you have a check on the consistency of this kind of information, like a backend, the check for taxonomy, for example. And then you need to add another person that needs to add all the other information about restriction of use, culture, growth, and so on. And my advice is also to keep track of the taxonomy. So even if a strain, like for the black aspergillus, for example, once they were Aspergillus niger, now they made a taxonomic revision and I have 27. No, they were 27. Now I don't know how many they are, because a few times that I don't work on that. But perhaps there will be some other term in taxonomy. So if you keep track of these changes in associated to your strain, but of course, for your catalog to be interoperable, you need to have the accepted nomenclature for the strain. So on the front end, you should make other people see the current name of the species, for example. Went out of topic, sorry.
Laura?
Yeah.
One topic that recently have in our collection is the accumulation of water in the plates and average lands when we store it in the fridge for active growth.
Because you store in plates.
And also in tubes in average lands. Do you have this similar problem there? Or
no, with tubes, no, because we use the cotton for our plugs. and hydrophobic cotton. It is a type of cotton that repel water.
Yes.
And we found it to be very useful because, for example, with the how do you say it? The cup, the silicone cup, with dry in times, so air and contaminants might enter. And with these cups with cotton, you can take in flask or tubes. Of course, the dehydration level is higher than if you close like with metal cups. So that's why you need to refresh your collection more often, but you won't have this kind of accumulation of water in tubes.
And you don't work with plates for
Do you know that there is this big problem with fungi and mites? So if I can avoid plates, I use plates just for isolation, when I get rid as fast as I can of that. Because in spring we have, even if you put all the effort that you have and you clean with everything that comes into your minds, the thermostats, you will have mites contamination, so cross-contamination problems. And there was one product that was very effective. I used it until 2004, and then it went super out of production because it was so toxic that you were not allowed to use. So yes, I would definitely avoid to use plates. That's it.
Thank you, Laura. Oh, wait, Silky has a question, sorry.
Sorry.
Yeah, we have a question from online. Yeah.
Yes, hello. Maybe a very, how you say, obvious question from my side, but what do you think about feasibility of keeping parasitic fungi in culture collections?
For the roundtable, like, well, we can discuss a briefly.
Just as a teaser, maybe like a first impression of like after like having seen like culture collections, and so yeah, from your experience now working with keeping fungal strains and bacteria strains, and
that's why I was asking you about some culture media, because it's easier, like for example, to preserve. The big problem with algae is they have vacuole that are very full of liquids that when you freeze dry, they make the cell explode. So with cyanobacteria and algae, you have this additional problem when you cryopreserve. That's why I was asking you about some media for the parasites isolation. But definitely this is what I want to make this roundtable, to try to see some solution on what can we do. Like I can give you my expertise, but I don't know what are the challenges in keeping. I imagine that. And so I was also thinking about some common experiments, like each laboratory can think about some methods we can try and see what's what's not.
Yeah. Well, then let's keep that then for the round table and to discuss further. Yeah, thanks.
Please just.
Thank you, Silky. I hear the comments here. You have